In recent years, advances in ancient DNA extraction and analysis have revolutionized our understanding of human history and evolution. One of the most groundbreaking discoveries in paleoanthropology has been the identification of genetic material from a previously unknown ancestor of modern humans. This discovery has not only expanded our knowledge of human lineage, but has also raised fascinating questions about population migrations, interbreeding between different human species, and the complex tapestry of our evolutionary past. The analysis of DNA from this mysterious has provided scientists with unprecedented insights into periods of human history that were previously shrouded in mystery, offering a window into the lives and movements of peoples who lived tens of thousands of years ago. The revolutionary field of ancient DNA analysis. Before delving into the specifics of this remarkable discovery, it is essential to understand the technological innovations that made it possible. The field of ancient DNA, aDNA analysis, has undergone a dramatic transformation over the past 2 decades. Scientists can now extract and sequence genetic material from bones, teeth, and other remains that are hundreds of thousands of years old. This ability to recover and analyze ancient genetic information represents one of the most significant breakthroughs in biological science. The journey toward this capability began in the 1980s, when scientists first successfully extracted DNA from museum specimens. However, the real revolution occurred with the advent of next-generation sequencing, NGS technologies, in the early 2000s. These powerful tools allowed researchers to sequence entire genomes from extremal, degraded and contaminated samples, something that was previously impossible. DNA is notoriously fragmented and damaged, with most samples containing only fragments of several dozen base pairs or shorter. Additionally, ancient samples are often contaminated with environmental DNA from bacteria, fungi, and soil organisms that accumulated over millennia. Modern ancient DNA labs employ sophisticated techniques to overcome these challenges. Scientists use highly sensitive equipment and specially designed clean rooms to prevent contamination. They employ specialized enzymes that can cope with the chemical damage characteristic of ancient DNA. Advanced bioinformatic methods allow researchers to distinguish authentic ancient genetic sequences from contaminating modern DNA and environmental genetic material. These technological advances have transformed ancient DNA analysis from a difficult, unreliable process into a powerful scientific discipline that yields consistent, reproducible results. The discovery of the unknown ancestor. The DNA of an unknown ancestor was identified through systematic analysis of genetic material from multiple ancient hominin specimens found at archaeological sites across different regions. Scientists discovered that several ancient human populations shared genetic sequences that did not match any previously known hominin species. This unexpected finding suggested modern humans, Neanderthals, Denisovans, and other known species were not the only players in the complex drama of human evolution. The analysis revealed that this unknown ancestor had contributed genetic material to modern populations, particularly in Africa and Asia. The genetic signatures of this ancient hominin were most pronounced in African populations, suggesting that the interbreeding between this unknown species and early modern humans may have occurred in Africa or in regions adjacent to the African continent. The discovery was made possible by comparing genomic data from multiple sources, ancient DNA extracted from fossil remains, genetic data from diverse modern populations, and information from known extinct human species like Neanderthals and Denisovans. The phylogenetic analysis, the study of evolutionary relationships, indicated that this unknown ancestor diverged from other human lineages hundreds of thousands of years ago. The timing of the split suggests that this species evolved separately from Neanderthals and Denisovans, following its own evolutionary trajectory in a region that may have been geographically isolated. The existence of this unknown ancestor indicates that human evolution was far more complex previously understood, with multiple species coexisting and occasionally interbreeding with one another. How scientists made the discovery. The identification of DNA from this unknown ancestor employed several sophisticated methodologies. One crucial approach involved analyzing the genomes of ancient humans from Africa, dating back 20,000 to 50,000 years. Scientists noticed that these ancient African genomes contained sequences that were distinct from any known human species, but showed clear signs of ancient interbreeding events. The sequences appeared to come from a species that had interbred with early African modern humans before gradually disappearing from the fossil record. Researchers used a technique called allele frequency analysis to track the presence of genetic variants inherited from this unknown ancestor. An allele is a variant form of a gene. By comparing allele frequencies across different populations and at different time periods, scientists could determine which genetic variants were inherited from unknown ancestors versus those that arose through other evolutionary processes. The discovery also relied on phylogenetic reconstruction, creating an evolutionary family tree based on genetic similarities and differences. By analyzing millions of genetic markers across the genomes of different populations and extinct species, researchers could model how different human populations diverged from common ancestors, and when these divergences occurred. The patterns revealed in this genetic family tree showed branches that couldn't be explained by known human species, pointing to the existence of previously unknown populations. Additionally, scientists employed machine learning algorithms to identify patterns in genetic data that might indicate admixture, mixing between populations. These computational tools could detect subtle signatures of past gene flow that might escape detection through conventional statistical analysis. By applying these advanced computational methods to large genetic databases comprising thousands of individuals from diverse populations, researchers were able to identify the genetic fingerprints of this unknown ancestor. Characteristics of the unknown ancestor. Based on the genetic analysis, scientists have been able to infer certain characteristics of this unknown ancestor. The DNA evidence suggests that this species had diverged from the lineage leading to modern humans approximately 500,000 to 1 million years ago, making it contemporary with, or even preceding, the emergence of Neanderthals and Denisovans. The length of time since divergence indicates that significant evolutionary changes would have accumulated, suggesting that this species possessed morphological and possibly behavioral characteristics that distinguished it from modern humans. The genetic analysis indicates that this species was capable of producing fertile offspring with early modern humans, suggesting that reproductive compatibility had not yet been completely lost, despite hundreds of thousands of years of separate evolution. This capacity for producing fertile hybrids suggests that the species had diverged recently enough that pre-zygotic barriers to reproduction had not fully developed. Such insights into breeding compatibility provide indirect evidence about the biology and genetic architecture of this ancient species. The genetic data also reveals information about population size and structure. The DNA signatures suggest that this unknown ancestor existed in relatively small, isolated populations, scattered across a particular geographic region. The genetic diversity within this species appears to have been limited compared to modern humans, suggesting that populations were small and experienced periods of isolation. This demographic pattern might explain why the species eventually disappeared from the archaeological record. Small, isolated populations are particularly vulnerable to extinction. The distribution of genetic material from this ancestor in modern populations provides clues about migration patterns. The concentration of this unknown ancestor's genetic material in certain regions suggests that interbreeding was localized to specific geographic areas. The persistence of this genetic material in modern populations indicates that hybrid individuals had reproductive success, and that their offspring interbred with both modern humans and possibly other human species. Implications for human evolution. The discovery of DNA from this unknown ancestor fundamentally alters our understanding of human evolutionary history. It demonstrates that the period from roughly 500,000 years ago to 50,000 years ago was characterized by the coexistence and occasional interbreeding of multiple human species. This finding transforms the narrative of human evolution from a linear progression to a more complex, branching pattern involving multiple parallel lineages. The existence of this unknown ancestor suggests that modern humans did not evolve in isolation, but were shaped by genetic contributions from multiple sources. The admixture between modern humans and this unknown ancestor indicates that the genetic diversity of contemporary humans reflects this complex evolutionary history. Understanding the geographic distribution of genetic contributions from this species helps scientists map out ancient migration routes and population movements. Furthermore, this discovery has implications for understanding which human traits may have been advantageous for survival in specific environments. If the unknown ancestor's genetic material persists in certain modern populations, it suggests that some of this genetic inheritance provided adaptive advantages. Scientists can now investigate whether specific genetic variants inherited from this ancestor are associated with adaptations to climate, diet, or disease resistance. Such research might reveal how ancient interbreeding contributed to the success of modern human populations in diverse environments. The discovery also raises important questions about species boundaries and human identity. In traditional taxonomy, species are defined as groups that cannot produce fertile offspring. However, the discovery of interbreeding between multiple human species suggests that human species boundaries were more fluid than previously thought. This finding encourages biologists and paleoanthropologists to reconsider the definition of species and how we categorize extinct organisms. Geographic and temporal context. The identification of the unknown ancestor's DNA has allowed scientists to map out the approximate geographic range in time. period when this species existed. The genetic evidence suggests a primary distribution in Africa, with possible extensions into surrounding regions. The genetic signatures are most prevalent in Central and West African populations, suggesting that this is where the most significant interbreeding with modern humans occurred. Temporally, the evidence indicates that this species existed for a substantial period, with interbreeding occurring during a window of time when early modern human populations were expanding through Africa and beyond. The genetic signatures become less apparent in more recent populations, suggesting that the unknown ancestor species disappeared from the fossil record, and that genetic admixture eventually was diluted as generations passed and populations continued to evolve. The timing coincides with a crucial period in human history, the era when anatomically modern humans were beginning their expansion out of Africa and into other continents. This timing suggests that the evolutionary pressures and environmental changes of this era may have been crucial to the survival of modern humans and the disappearance of other human species, including this newly discovered lineage. Scientists now understand that at various points in the past, as many as 4 or 5 different human species coexisted on Earth. We know of Homo sapiens, modern humans, Neanderthals, Denisovans, and Homo floresiensis, the hobbit. With the addition of genetic evidence for this unknown ancestor, the picture becomes even more complicated. The phylogenetic relationships between these species are now better understood through genetic analysis. The unknown ancestor appears to have been more distantly related to Denisovans and Neanderthals than modern humans are to either of those species. This suggests that the unknown ancestor represented an earlier branch in the human evolutionary tree, diverging before the lineage split that eventually gave rise to Denisovans and Neanderthals. Interestingly, there is genetic evidence suggesting that Denisovans themselves interbred with populations that may have encountered the unknown ancestor's descendants. This creates a complex network of genetic inheritance where modern humans carry genetic material from multiple ancient species, each contributing small but measurable portions of our genome. The unknown's ancestor's DNA adds to this mosaic of human ancestry, making us truly composite organisms in terms of our genetic heritage. Challenges in ancient DNA research. Despite the remarkable advances in ancient DNA analysis, significant challenges remain. The unknown ancestor's DNA was discovered not through finding preserved remains with extractable DNA, but through detailed comparative genomic analysis. In many cases, the fossil record does not preserve bones with extractable DNA. The discovery relied on inferring the existence of an unknown ancestor from patterns in genetic data, an indirect method that requires careful interpretation. Prevent contamination with modern human DNA. Archaeological sites may have been disturbed by multiple visitors over centuries, must carefully distinguish authentic ancient sequences from contaminating DNA. Another significant challenge is the sparse nature of ancient DNA sequences. Most ancient samples yield only fragmented DNA with an average read length of just 50 base pairs. To construct a complete picture of an ancient genome, scientists must sequence millions of fragments. This requires substantial financial investment and computational resources. Not all laboratories worldwide have access to the equipment and expertise needed for rigorous ancient DNA analysis. Additionally, interpreting genetic signals requires careful statistical analysis. Scientists must distinguish genuine signals of interbreeding from artifacts that might arise from technical issues or methodological shortcomings. False positives, incorrectly, identifying genetic signals where none exist could lead to erroneous conclusions about ancient populations. Peer review and replication studies are crucial for validating discoveries in this field. What this means for modern human genetics: the genetic material inherited from the unknown ancestor is now part of the genome of millions of people today. The percentage of DNA inherited from this species varies among individuals and populations, but typically represents between 1 to 5% of the total genome in the most affected. Understanding this genetic inheritance has practical implications. Geneticists can now account for the unknown ancestors' contribution when studying genetic disease susceptibility, drug metabolism, and other phenotypic traits in modern populations. Certain genetic variants inherited from the unknown ancestor may influence traits ranging from skin pigmentation to metabolic efficiency. Researchers are investigating whether any of these variants provide adaptive advantages in specific environments. For medical genetics, this knowledge is particularly important. Some genetic diseases show different prevalence rates in different populations. Understanding the contribution of ancient admixture events to genetic diversity helps medical researchers better understand disease susceptibility and develop more effective treatments tailored to genetic ancestry. The study of ancient DNA has become an important resource for human geneticists seeking to understand the evolutionary origins of human genetic variation. Future directions in research. The discovery of the unknown ancestor opens numerous avenues for future research. Scientists are actively searching archaeological sites for fossil remains that might belong to this species. If remains are found with sufficient preservable DNA, direct genetic sequencing might yield far more. Information then can be inferred from patterns in modern genomes. Such discoveries could revolutionize our... Researchers are also applying increasingly sophisticated methods to extract maximum information from modern genetic data. New computational approaches continue to refine our ability to detect ancient admixture signals and estimate when interbreeding events occurred. As genomic databases continue to expand to include thousands more individuals from diverse populations, especially underrepresented populations, our picture of ancient admixture events will become clearer. Ancient environmental DNA analysis offers another promising frontier. Scientists can analyze DNA preserved in soil and sediment. Viewing human evolution as a straightforward march toward modern humanity, we now recognize it as a complex web of evolutionary branches, population movements, and genetic exchanges. This more nuanced understanding reflects the reality of biological evolution, a process full of contingency, adaptation, and occasional interactions between lineages. This discovery also highlights the importance of genetic data in complementing archaeological and paleontological evidence. While fossil records often provide incomplete information, DNA analysis can reveal population movements and relationships that leave no trace in the physical record. Conversely, fossil evidence places genetic findings in temporal and geographic context, helping scientists interpret what genetic signals mean in terms of actual prehistoric populations and their lives. The study of ancient DNA has also democratized paleoanthropology. Scientists no longer need to discover complete skeletons or rely solely on morphological analysis. DNA from small bone fragments or even teeth can yield rich information about populations and their histories. This opens research opportunities in regions and time periods where fossil evidence is sparse. Conclusion. The discovery of DNA from an unknown ancestor represents a milestone in our understanding of human evolution. It demonstrates that the evolution of our species was shaped by interactions with multiple other human lineages, each contributing to the genetic heritage of modern humans. This finding transforms the narrative of human origins from a simple story into a complex saga involving multiple players and unexpected twists. As technology continues to advance and our databases of genetic information grow larger and more diverse, we will undoubtedly make additional discoveries about human evolutionary history. Each new insight brings us closer to a complete understanding of where we came from and how we became who we are. The unknown ancestor, identified only through genetic analysis, reminds us that much of human history remains hidden, waiting to be discovered through the clever application of modern science to ancient genetic material. The more we learn about our evolutionary past, the better we understand ourselves and our place in the natural world.